There are 4 major phases that happen with contraction. So actually causing the shortening of your muscle fiber and then the relaxation of your muscle fiber. First phase is excitation. So this is where your action potential from your nerve is sent down that neuron to signal your muscle fiber. Then you have your excitation, contraction, coupling, which activates the myofilaments, preparing them to contract. Then you actually have your contraction stage where your sarcomere shortens with the overlapping of your myofilaments, so developing tension and then shortening that sarcomere, and then relaxation, the last part where your stimulation ends and the sarcomere, which is part of your muscle, returns to its original resting length. So looking at the pictures, with our nerve signal traveling down the axon of our neuron, once that action potential, again also traveling the surface of the nerve's plasma membrane, it will trigger voltage-gated calcium channels to open, causing calcium to enter in the cell. That calcium then causes the synaptic vesicles filled with our acetylcholine ligand to attach to the plasma membrane and release acetylcholine into that synaptic gap. Once it releases, it binds to binds to those acetylcholine receptors on those junctional folds, which allow for more space or more area for our acetylcholine receptors on the plasma membrane. So these acetylcholine receptors bind acetylcholine. They worked as a gate, so a ligand gated ion channel for both sodium and potassium. When the sodium and potassium go down their concentration gradient, it starts off the first process of the action potential, so it changes the membrane potential just enough to cause the action potential. So getting that first trigger, that action potential starts, it travels along your plasma membrane into the T-tubule. When the action potential travels along the T-tubule, causes the release of calcium from our sarcoplasmic reticulum, so those terminal cisterns there, part of this triad structure. The calcium is then released. It moves from area of high concentration to lower concentration where the myofibrils are, to bind to the troponin regulatory molecule. So that's this little guy in yellow. Calcium binds. It causes a conformational change. So the tropomyosin moves off of those active sites on actin. So there's little holes here. Those provide the binding site for the myosin protein, which is the proteins that make up your thick filaments. So for the actual contraction, this is where your ATP comes into play. So you need the hydrolysis of ATP, so going from ATP, our 3-phosphates, to ADP and extra phosphate. So ATP broken down into ADP and excess phosphate group. When that hydrolysis happens, it causes a conformational change in the myosin protein. So at the first part of contraction, causing the head of the myosin to cock back, so it's in position now to bind to those active sites on actin, helping to create the myosin-actin cross bridge. So this is your recovery stroke. For your power stroke, so this is the part that's going to actually cause your contraction, the ADP and phosphate group remove off of your myosin head, causing another conformational change. When that conformational change, you go from the cocked back myosin head, which is bound to our actin site, it pulls on those actins, so on those thin filaments, pulling towards the center of your sarcomere, causing your power stroke. So as that pull happens toward the central part of your sarcomere, causing an increase in overlap of your thick and thin filaments, shortening the sarcomere, shortening the rest of your muscle, and causing the contraction. So again, this is your power stroke. Then we have another ATP molecule that binds to the myosin head, causing the breakage of that link between the myosin head and your actin subunit. And then in order to cock back that myosin again, so needing that recovery stroke, so causing the relaxation for your muscle, ATP is hydrolyzed again to cock it back and allow for your sarcomere to go into the recovery stroke, so lengthen back out again. Next phase is relaxation. So part of relaxation is stopping the acetylcholine ligand from continuing to signal the muscle cell to contract through its binding to the acetylcholine receptor. So once your acetylcholine releases from your acetylcholine receptor, the enzyme acetylcholine esterase, so ACHE for short, breaks down this acetylcholine, and then those bits are taken back up into your neuron, then trafficked all the way back up to the neurosoma, or the main cell body of that neuron, put back together, and then trafficked back down again that axon, back to the axon terminal. Other phases of relaxation are the returning of calcium to those terminal cisterns, part of the triad. So returning it back to your terminal cisterns, helping to sequester your calcium and make it ready for the next contraction. So one of the very important things about needing calcium inside your muscle cells, so having enough calcium running in your body, without the calcium ion, troponin returns to blocking the active site, so it removes off and gets reabsorbed. So as long as you continue to have calcium to remove the troponin off of, binding troponin, removing the tropomyosin off of those active sites, as long as you still continuously have enough calcium, you're still able to continue to have contractions. So here is a sarcomere during pre-contraction, so haven't got the signal yet to contract. So once that acetylcholine binds, makes that action potential go, release of calcium on the troponin molecule, the tropomyosin molecule then moves off those active sites on actin, allowing for the myosin head, which are little purple sticks, off of our thick filaments in this little picture, able to make that myosin-actin cross bridge. Then the release of the ADP and phosphate group causes that conformational change in the myosin head, causing the power stroke. So shortening of your Z disc. So here we have two sarcomeres, have our thick filaments. Of course, I didn't draw the elastic filaments that run through it, but they're there. So we have a little bit of overlap between our thick and thin filaments. So once we get the signal to contract, it shortens the sarcomeres. So here we have the power stroke for our myosin heads. Our sarcomeres have shortened. So they help to pull on each other to cause the shortening of the entire muscle to cause your contraction. Then for relaxation, the sarcomeres return back to their original length. In order to record the activity of muscle, you can do this using a myogram. It's a chart of the timing and strength of a muscle contraction. So for a muscle contraction to happen, again, you need your muscle to go through a threshold, so a change in the membrane potential enough to get to the point to open those voltage-gated ion channels and causing the action potential. So again, our action potential is the quick change in membrane potential with our depolarization, with our sodium rushing into the cell, our quick repolarization with the potassium moving out the cell. So there's your action potential, travels along your cell, eventually signaling the shortening of your sarcomeres through increased overlap of your myofilaments. When this happens, it causes a muscle twitch, so it's a quick contraction and then later relaxation of your muscle. based on a stimulus that is at least at thresholds or higher. So you need enough of stimulation to actually get the contraction to go. So here we have a cartoon of a myogram. So you have your initial stimulation here. There's a latent period where you're generating tension in your muscles, so getting everything ready to contract. Then you have your actual contraction phase, and then the slightly longer relaxation phase to return back to the original muscle tension. So here's muscle tension on our y-axis, and then this happens over time through our x-axis. So for the stimuli, again, they have to be above threshold to actually get a muscle twitch, so the contraction and relaxation of a muscle. So up top, we have the relationship of looking at our stimulus voltage, so each one showing an increase in voltage for a stimulation, dashed line being threshold. Down here are the resulting contractions or not, or muscle twitch of that muscle. So the first two are below threshold or sub-threshold, so they're not actually able to recruit muscle units. So let's zoom this in. So both of these sub-threshold stimuli are too low to recruit muscle fiber units, so therefore you're not going to have a contraction. So seeing our increase in our voltage for our stimulus, first one reaching threshold, able to recruit a muscle unit, and then increasing levels of muscle units as we increase the voltage of our stimuli. Once we get to the point of recruiting all of our motor units, then we are at our maximum level for contraction. So looking at our contraction, our chart at below, so the first two, there's no contraction, there's no tension in that muscle because the stimuli are below threshold. Then we have a little small one right at threshold, a little bit more with increased stimuli, until you get to the point of where all of your muscle motor units are recruited. When that happens, then you're at maximum contraction. So you're not going to get a stronger contraction by giving someone increased voltage. So by increasing your voltage helps to recruit more motor units, giving you a stronger stimulus. Due to a stronger stimulus, giving you increasing the level of recruitment of your multiple motor units. Once you get to all of your motor units being recruited, you're at maximum contraction. So no matter how much more you increase your stimulation, you're still not going to have a stronger contraction. The timing of your stimuli also causes differences in your muscle contraction. So in our pictures, we have the same amount of stimuli, so same voltage, all of these being above threshold. However, the myograms look different because of the timing. So for a low frequency stimuli where your muscles have enough time to fully relax, you'll have muscle twitches that are of identical strength. So peak being at the same level for each muscle contraction. However, at higher frequency stimuli, so same amount of stimuli, so same voltage level, except for they're so quick in their stimuli, that quick in their timing, that your muscles don't have time to fully relax. So here's our baseline, then a little bit higher up here is the partial relaxation that happens between the muscle twitches. So when you get the same stimuli strength, it causes a same height of or amount of twitch, but it's a little bit stronger because you're still partially relaxed. You're only partially relaxed, so you have already a little bit of extra tension on your muscles. So it's kind of like stairs, where wherever you are, even though you're only going to make the same amount of height adjustment to get to the next step. Here, because you're already slightly above where you started, you increase the level of tension. So each of these, because you're not being able to relax fully, able to increase the tension till you get to what's called a incomplete tetanus, so a temporal wave summation, so adding up of your waves based on the quickness of time, so a temporal, and continues to build up in strength. For stimuli that's unnaturally high in frequency, so very, very quick stimuli, so still the same voltage, but the timing of it is so quick that, again, you have partial relaxation. Except for here, instead of allowing at least some relaxation for our temporal wave summation, the relaxation is so short because you're already getting another stimuli that you quickly build up into what's called a complete tetanus. And this is also called a fused tetanus, where there's a steady contraction and your muscle, it just stays contracted. So it has no time to relieve the tension in that muscle. Once you stimuli a muscle or give muscle a stimulus, you don't immediately get a contraction. So you have to build up tension enough to actually cause movement. So there's 3 types of contraction. You have your isometric contraction, where you're building tension in the muscle, but the muscle doesn't shorten. So here we have the lady lifting a dumbbell or barbell. No, dumbbell. So there's tension in the muscle, but there's no movement. That's an isometric contraction. So let's say if we took the took the dumbbell away from the lady, and then gave it back to her, this muscle would have to have tension in order to stay in the same level of shortening to continue to hold that dumbbell in that position. In order for her to move the dumbbell, you have to be able to shorten the muscle, pulling on those bones to cause the movement. So for our bicep curl, that's our isotonic concentric contraction. So for this concentric contraction, actually causing the movement based on the shortening of this muscle. So increasing the tension, where get to the point of your tension remains constant, but your muscle shortens. So shortening of your muscle causing the upward movement here. For relaxing of our dumbbell curve, so moving our dumbbell down, is isotonic eccentric contraction. So your muscle lengthens in how long it is while maintaining tension. So to see this in a chart form, so again, your isometric phase is your beginning, where there's no change in the tension, but no change in the length of your muscle, but the muscle tension rises. Once your tension overcomes the resistance of the load, so holding up that dumbbell, then your tension levels level off, and then there's a change in your muscle tension. So for example, here over time we have an isometric phase where there's still tension, still building tension, until you get to the point of being able to shorten your muscle. So you've got to the point of you're at full muscle tension, so you're not changing your muscle tension. Now you're able to change the muscle length because there's enough tension in your muscle to overcome the resistance of the load. So basically the weight of whatever you're trying to lift or move. So even if you're just moving your arm, there's still a little bit of weight in your arm. So without, for example, a dumbbell, there still needs to build up of tension before you actually are able to move your arm up. Not much of a tension build, but still has to build that tension up beforehand. So again, the first phase, isometric, where there's no change in the length of our muscle, switching until your tension has built up enough to actually cause movement, either through a concentric contraction to shorten the muscle, or through a eccentric contraction to lengthen the muscle and still be able to resist the weight. So instead of letting that weight just drop down due to gravity. So controlling of the movement. Moving on to powering of our muscles. So our muscles are powered based on our ATP molecules. So that's our energy molecules for all the cells in our body. So all of our muscle contractions need ATP. Again, going back to earlier when we talked about contractions, ATP is necessary for not only contraction but also relaxation to make that conformational change in the myosin head due to the hydrolysis of ATP. So going from ATP to ADP and that excess phosphate group. So in order to make ATP, you have to have plenty of oxygen as well as an organic energy source. So either through glucose from the glycogen in the in those storage molecules inside of muscles, as well as other cells, as well as from fatty acids. So next semester we'll actually cover how fatty acids are able to be used to make ATP. So there's two pathways for making ATP based on the level of oxygen. So if there is an absence of oxygen, your cells are still able to make ATP through a process called anaerobic fermentation. There's only little ATPs made, actually only 2 molecules of ATP is made, and one of your intermediate molecules, starting, for example, from glucose, going from pyruvate, which is that intermediate molecule, fermented into lactate. The other type of making ATP is a whole lot more efficient. So it produces a lot of ATP. So instead of only 2, it's actually 32 for aerobic respiration. Does not make lactate, which needs to be disposed of by the liver. So actually your molecule gets so broken down that it breaks down into carbon dioxide and water. So body's able to use the water, and then your body just breathes out the carbon dioxide. For aerobic respiration, you need a constant oxygen supply, and that helps to power the oxidation reduction reactions that happen during that whole process. Again, this is something that we'll cover next semester. So during the process of exercise, there's changes in how our muscles make our ATP. So starting off, our muscles have enough glucose in the form of glycogen as well as plenty of oxygen on those myoglobin molecules to make ATP through aerobic respiration. Eventually, you switch into what's called the phosphagen system, where there's just some switching around of phosphate groups to continue make ATP. Then eventually, you switch into the glycogen lactate system, so you're kind of starting to run out of oxygen because the myoglobin had given up their oxygen to make ATP. So switching to anaerobic fermentation for a little while. Eventually, your aerobic respiration picks up again because you're able to bring in that oxygen through breathing, therefore powering up the aerobic respiration again, able to make more ATP through aerobic respiration. Moving on, so not all muscle fibers behave the same. So there's 2 main types of muscle fibers. You have your SlowTwitch muscle fibers and your FastTwitch muscle fibers. So your SlowTwitch muscle fibers, also called your slow oxidative, red, or type 1 fibers. These muscle fibers are more so for in distance. They resist fatigue using aerobic respiration to make ATP. So these slow twitch fibers are in muscles that help to maintain posture. So helping us to sit upright, our core muscles helping us to hold in our stomach and hold in our different intestinal gastro organs, the muscles of the back to help us stay upright and keep our posture. So we're supporting our back. So anything that helps us to stay in position. So for example, some exercises that require balance and you're just staying in position, you're using a lot of these slow twitch muscle fibers. So for example, yoga is one way to exercise these muscles. These slow twitch fibers have a lot of mitochondria, so to make sure that they have plenty of ATP to resist going through fatigue, lots of capillaries so that they have plenty of oxygen being sent to those cells, and then myoglobin, plenty of those that allow for storage of oxygen, giving these muscle fibers their deep red color. These fibers are a little bit thinner. They're grouped in smaller motor units for more precise movement, so those slight adjustments that you need for making sure that you have your balance and posture. So for example, like standing on one leg, needing slight adjustments in your posture as you're trying to maintain that position. Your other type of muscle fiber are your fast twitch, also called your fast glycolytic, white, or type 2 fibers. So this type of muscle fiber is for more of a quick response. So these are very quick, powerful muscles. So for example, in the eye and hand, so blinking, moving stuff quickly in the hand to do stuff with, as well as your gastrocnemius in the calf muscle and your biceps brachii. So that kind of the muscle that you work out a lot in the gym probably. So make sure it's nice and big and looks all cute. So that's the one that's in between your elbow and your shoulder. A lot of people like to flex that one. So again, very powerful muscle, able to quickly move. So for example, like a lot of your fast sports, tennis or basketball, you see those athletes having ginormous, very large calf muscles, particularly, for example, Serena Williams, very large calf muscles, but necessary for tennis. They use glycolysis and anaerobic fermentation for energy. Again, they're going to move very fast, very quick shifts in their contractions. So just makes enough ATP for whatever they just need it for. And then they'll relax, and so you don't need that ATP anymore. Fibers are usually thicker and stronger with large motor units. So one neuron, ton of cells that it targets so that you have a quick contraction, able to have those quick motions for these fast twitch fibers. So all of your muscles contain a mixture of your two fiber types, but one usually predominates in that muscle depending on function. So for example, going back to your calf muscles, mostly predominating with your fast twitch fibers, but still having some of your slow twitch ones, for example, just for standing, maintaining your posture while standing. Your fiber type varies between different individuals. So for example, for someone who's like Usain Bolt, fast runner, is going to have a lot of those fast twitch muscle fibers in his muscles to be able to quickly respond for running and making sure that he, wins the race. For someone who does like more yoga or like ballet, for some of it, they might have a little bit more on the slower twitch, so being able to keep their balance and maintaining particular positions. Now finally, moving on to our cardiac and smooth muscle. So our cardiac and smooth muscle have certain properties that they share. So their cells are still called myocytes, but they have only one nucleus, unlike your skeletal muscle that has multiple nuclei. Sometimes your cardiac muscles have their own name called a cardiomyocyte, still same type of cells. Both your cardiac and smooth muscle are involuntary, so we cannot consciously control them, so we cannot tell our heart to slow down or speed up. We also can't tell the smooth muscle that makes up our hollow organs on the inside of our body to contract whenever we want to. So again, involuntary, unconscious control for it, so our brain can't control it. Both your cardiac and smooth muscles receives innervation, so receives nerve signals from the autonomic nervous system, so not from a somatic motor neuron that is controlled by our brain, but through our autonomic nervous system that is controlled by the brain stem. So properties of our cardiac muscle: contracts with a regular, consistent rhythm, and this is due to pacemaker cells within our cardiac muscle that causes them to contract. So these pacemaker cells set off your cardiac muscle, so it actually doesn't need nerve system innervation, so it doesn't need a nerve signal to tell it to contract. It does it by itself. The innervation from the autonomic nervous system helps to adjust your heart rate, so getting it to speed up when you need to go through some kind of action phase. So like if you're running away from a lion or from a dog, or helping you to calm down when you're trying to go to sleep and helping you to relax. So adjusting your heart rate, but it doesn't tell your heart to go. Again, heart, of course, it pumps blood all the time, so it works nonstop, so therefore resistant to fatigue, having plenty of oxygen and glucose in order to make ATP to continue to contract. One thing that's special about your cardiac muscle is that they contract in unison, so all your chambers will do so through some of these structural features on your cardiac muscle. So through those intercalated discs, allowing that signal to go from cell to cell to cell, telling all of them at roughly the same exact time to contract to themselves. So these contractions have to last long enough to expel blood. So our heart contains 4 chambers, so 1, 2, 3, and 4. Actual, better way to say 1, 2, 3, 4. And that's the order how the blood flows. So each of these chambers will contract at the same time so that they're moving blood in the correct direction through the heart. Things that are unique for your cardiac muscle is that it's striated just like your skeletal muscle. So there's still a overlap of those thick and thin filaments, but your cardiomyocytes are shorter and thicker and branched, unlike your skeletal muscle. Cardiac muscle communicates between cell to cell through these intercalated discs. So what you learned from chapter 5 in the tissues. So these discs work as not only electrical gap junctions to be able to send that nerve signal from cell to cell so that they contract in unison, but also as mechanical junctions to keep the cardiomyocytes together. With your cardiac muscle cell, so for example, let's say you have a heart attack. You're no longer receiving oxygen to that cell, and so therefore those cells become damaged. To repair this damage, the cardiac muscle does it through fibrosis. So that patch of cells are no longer going to work like they should. Again, your cardiac muscle doesn't need nerve stimulation to cause the muscles to contract. So the pacemaker cells that are inside the cell help to set the heart to cause it to contract. So traveling through those intercalated discs and be able to contract on its own. So it's autorhythmic. For your smooth muscle, it's called smooth because it does not have striations. So for your nerve supply for your smooth muscle, instead of being a direct contact between your neuron and your muscle cell, some of your smooth muscle actually lacks a nerve supply. It gets its signal from varicosities. I'll show you in a picture in a second. That contain synaptic vesicles, and they kind of work like a sprayer on a farm field, where instead of having directly right close to those receptors, it just sprays those ligands or the chemical messengers across multiple cells, and then you just kind of cross your fingers that these cells get the signal to contract. Smooth muscle is able to go through mitosis, so able to repair itself, and go through also hyperplasia, regenerates very well. Because of this, smooth muscle moves slower than skeletal and cardiac muscle, so it takes longer for it to contract, but it's able to do so without fatigue. So thinking of how long it takes your stomach to digest stuff. So here's a picture of a type of smooth muscle. In a single unit smooth muscle, there's also another type of unit that's a little bit more direct, but we're just focusing on this type. So here's our nerve in yellow. And the little bulges are varicosities, so they just release the neurotransmitter close to the cell, and then eventually that neurotransmitter going from high concentration to low concentration will eventually find its way to the cell and causes